Okay, so again, my name is Tim Meeker. I'm hoping to open here the Pain Perception and Attention Integrative Neuroscience Lab, or the PAIN Lab. And current projects which people could be involved in are neuroimaging meta, and I'm going to go through basically what I mean by each of these. Neuroimaging meta-analysis, racial and sex differences in brain responses to painful stimuli, which I have already the data for, just never been analyzed. Brain activity at rest, which we can get an enormous amount of data for, depending on our ability to analyze it. And then behavioral responses to painful stimuli. So none of these things have been fully worked out in the data sets that I have, and they don't have a very long history of looking at research. So an additional question that I'm looking at is, how does pain sensitivity and somatization, which somatization is basically the tendency to think that something you feel in your body, like a somatic event, internal event, is related to a disease. Affect resting EEG and laser evoked potentials. I won't get too much into that today, but basically these are potential brain potentials that we measure on the scalp of people while they experience a painful stimuli. We also have quite a bit of data on fear conditioning to painful stimuli. And then I've done quite a bit of survey work, and I'm interested in survey work generally. So if it comes down to it, you need to do a survey, I can help with that as well. So on to the neuroimaging meta-analysis. Basically, what this does is it takes, and I'm working on one on sharp mechanical pain that has never been published, or was published once like 20 years ago, 10 years ago, which makes it extremely outdated by neuroimaging standards. We can use software where we can analyze previous reports of any particular experimental design and render a group map of what parts of the brain participate in that activity. And frequently you can publish that as a manuscript. My example here will be sharp mechanical pain. I had an undergraduate at Johns Hopkins and an undergraduate here who both worked on this project. Wow, I really blew that up, didn't I? So this is a look at the brain's reaction to non-painful mechanical stimuli in healthy people. And what you can see is, this is an area called the posterior insula in the parietal operculum. And then you have areas of the somatosensory cortex and other areas of the somatosensory cortex, and a little bit of reduction in activity in an area called the posterior cingulate, or precuneus, I should say. That's near the posterior cingulate cortex. Now, this makes perfect sense because, you know, a majority of these stimuli were delivered on the right hand, which this is the, let me see, is this the right brain? Okay, this is neurological, so that's one thing you have to learn, is that a lot of people that show brain images will flip them because they're either looking from the patient, the radiological standard is looking straight on top of the patient's head, whereas the neurological standard is to look at the patient from in front of you, so right and left will be flipped. Right and left is not flipped here. So this sort of makes sense. The parietal operculum, which is also known as the second somatosensory cortex, is involved in feeling mechanical, tactile mechanical stimuli. Now, if we look at painful mechanical stimuli, we get just a few more really additional areas. Interestingly, and nobody's looked at this, so if anybody really wants to dig into this, you may have like a really awesome set of papers and maybe a career even. We have deactivation in the visual cortex, which nobody knows what means, though it's been recorded both in humans and in animals. And then we have activations in anterior insula, which are these top parts here, and then the posterior insula, which is more the bottom part, where there's most of the activation. And again, this recapitulates the same story that has been told for about since 1996, 7, something like that. So that makes sense. Now, the question is, what areas, if any, are specific to painful mechanical stimuli compared to these non-painful mechanical stimuli? And we do this basically with a big t-test. It's a huge t-test. And we have all these methods to control, but the areas that are really interesting to me are the PAG, which is down here at on the very first slice. The dorsolateral prefrontal cortex, which is left dorsolateral prefrontal cortex, or right dorsolateral prefrontal cortex here, sorry, on slide 34. And then on slide -4 at the bottom, you can see cingulate cortex. What do you think's in common about all these areas? They're regions of the brain that release opiates naturally. So we have strong activation only during painful stimuli, or preferentially during painful stimuli, to areas of the brain that are involved in modulating pain. And so that's really where a lot of my work is focused, is looking at that network in the brain that modulates pain. Because what I want to do is I want to modulate that network in people with chronic pain illnesses, allow it to work better, because it doesn't always work as well. And let's see, what else do I have here? So if you look at the patient response, places where patient responses are greater, so these are patients with chronic neuropathic pain, you see that they have greater activation in the right dorsolateral prefrontal cortex, which is pretty normal. You see that, right amygdala and right dorsolateral prefrontal cortex. The amygdala is on the -14 slice, and then the dorsolateral prefrontal cortex is on the 18. And you get that over a group of, this is hundreds of patients and about 20 published studies. So you can do something similar to this, looking at anything that anybody's looked at in neuro, because what we can do is we can extract that data. For example, ask the question, well, what does the brain look like when people are empathetic to someone's pain when they're not, versus when they're not empathetic to someone's pain? And yeah, so you know, this is a paper that needs to be published pretty soon. And we've gotten pretty far with a couple of undergraduate students that worked on this for about 18 months, I think. And you know, this actually is going to lead me to write parts of a grant to work on that. So the second set of questions involves, are there racial differences when the brain experiences pain? Right now, there have been 3 publications looking at this. One was in Nature Behavioral, one was in Pain, this one, by Janelle Lepson, who's a friend at Johns Hopkins. And basically what she's looking at is she's looking at the binding potential of carfentanil. And if you don't know, fentanyl is extremely powerful, 50 to 100 times more powerful than morphine. Carfentanil is used to anesthetize elephants. That's the only real therapeutic use it has. But because it has such a really strong binding to mu-opioid receptors in the brain, we can look at areas of the brain and ask the question, well, what areas of the brain have differential binding based on either a painful stimulation or, in this case, based on race. And this is just looking at non-Hispanic whites and non-Hispanic blacks. And what we found, or what they found, was that there was, I gotta double check this. So bind tech experiments are always counterintuitive. So you gotta make sure you're doing the right, saying the right thing. Mouse. Okay. So what I'm gonna say is, I can't move my cursor. Okay, give me a second, sorry guys. My cursor just decided to leave the building. There it is. So presentation mode. I want to share. Come on now. That's what I want to share. I'm going to reverse. All right. Okay. So, like I said, this is kind of counterintuitive. On the bars, what you see is that all those areas circled in red, except for the PAG down here, which I'm interested in. So you have the dorsolateral prefrontal cortex bilaterally, you have areas of the cingulate cortex, and then you have an area called the ventral striatum, which is basically also called the nucleus accumbens, which is involved in reward motivation. In all of those areas, there's higher carfentanil binding in non-Hispanic blacks than there is in non-Hispanic whites. So what that tells us is that there's relatively lower occupancy of those receptors for some reason. Nobody knows why. We just discovered this, like I said, 2 years ago. And this is counterintuitive to what they expected. So this is a novel result, but it's kind of counterintuitive. Now, when we look at brain responses to painful stimuli, these are areas where non-Hispanic blacks have a greater pain response to heat stimuli, painful heat stimuli, than non-Hispanic whites. And what we see again here is bilateral DLPFC, dorsolateral prefrontal cortex, and also that nucleus accumbens region again. So it's also the ventral striatum, as we saw in the last slide. So this is interesting because these are opiate releasing pathways. We have higher activity in those areas, but during pain, there's lower occupancy of the receptors. So what does that mean? That means that for some reason, that system seems to be less effective. The nice thing about that is that now we can use non-invasive brain stimulation to enhance the effectivity or the efficaciousness of those brain areas, theoretically. I mean, this is a long-standing project. And then another thing that this paper looked at was nucleus accumbens activity is not positively correlated with the frequency of discrimination that people experience, except for in African Americans. So this is sort of like what they thought was sort of like they were trying to figure out exactly why this happened. Do they have enhanced discrimination or frequency, or are they receiving enhanced discrimination? These are healthy people, so otherwise healthy people. And so it seems like there's an additional burden on the brain to deal with painful stimuli. So changing gears a little bit, this is sort of basically a menu for what we've done. So I did all my studies in Baltimore. I have at least 60 subjects that we've run through this type of experiment. And so we do a structural brain image. We look at resting state brain. We look at something called arterial spin labeling, which basically looks at the blood flow in the brain, and it's actually like quantitative rather than fMRI is not quantitative. It's relative to when you get in the scanner because there's all these artifacts that go on that we have to control for. But this is a particular scan that allows us to be more quantitative. In the same group of subjects, we apply these pinprick probes, which are basically painful, sharp, painful. It's like a needle, but it doesn't penetrate the skin. I applied capsaicin, which basically turns most heat or warm stimuli into painful heat stimuli for most people, but not all. And then we took another type of structural scan to look at the white matter tracts in the brain. And we took the resting state scan during the painful stimuli. We did pain ratings. We did an ASL scan during the painful stimuli. And then we did the pinprick probes again while the participant had capsaicin on their leg. So this produces something called central sensitization, where the spinal cord basically recognizes the painful or the stimuli, and they say, this is much more painful. So you get secondary hyperalgesia and central sensitization when you burn yourself. The area around the burn becomes painful as well. So we have this on, depending on the data set, 40 to 60 people, and we just have started publishing from it because it's been a very large. One thing that we found that I found was particularly interesting is we're looking at the connections between regions of the brain network during pain and during pain-free. So the pain-free state is in blue, all that data is in blue, and then the pain state, tonic pain state is in red. What you can see is that this area from the part of the ACC that you saw lit up and having higher activity in African Americans has greater connectivity, or its connectivity is related to the pain intensity that they're experiencing during this, basically this capsaicin pain, which lasts 45 minutes. It's related to their pain intensity only during the painful stimuli, but not during the resting state. Now, what's really weird is that if we look at in the brain stem, and there's brain stem projections to the periaqueductal array, and we look at the normalized pain intensity there's only a relationship before they experience the pain, before the subject, so we thought that's very interesting. These were the only relationships out of like 30 that we looked at, and all of these were in the literature, and everybody was, but the problem is, one of the big issues with neuroscience right now, especially neuroimaging, is that people used to do studies on 8 to 12 people and think that that was sufficient. So this is 50, and we were basically not able to replicate a lot of things. And so, it was a lot of work, but it was important because we know that the things that we didn't replicate probably aren't related to the science of pain. Let me see here. So again, another data set that I have, this is pinprick probe data. We can look at areas where pain intensity is correlated with the brain response. Again, we see the PAG. This is something called the temporal parietal junction that I'm not fully aware of, don't fully understand yet. We have the anterior cingulate cortex. We have this area, which is -4, which is the paracentral lobule. So when the brain curves, right? So you have the two lobes of the brain, human brain. All of the area on the cortex that represents the face, hands, torso, arms are basically on the surface. This paracentral lobule bit is basically from the knee down. So we were stimulating on the leg because we put them in this large magnet. If you've ever been in a big MRI, we put them in this big MRI and we couldn't really reach anything else. So we use the leg. So you can see that, the pain intensity correlates positively there. All of this has been shown before, but not in as big of a data set. And then, we get the posterior insula, left posterior insula, the thalamus. All of these are areas we would expect to see that would be related to pain intensity. So let's see what else I decided to add last night. So that was published in this work. This is a horrible title. It's the first paper I wrote, so I'll forgive myself. Non-invasive motor cortex neuromodulation reduces secondary hyperalgesia and enhances activation of the descending pain modulatory network. So that's a mouthful, but what it means is that we were able to use this non-invasive brain stimulation for some reason of motor cortex. Don't know why, nobody knows why. And we were able to enhance the activity in other brain regions. So, in particularly the responses to these painful stimuli in regions of the brain that release opiates. So, a little bit about the method. Motor cortex stimulation, basically we have two very large electrodes that pass 1 milliamp of current. And if you want to know what 1 milliamp of current feels like, it's less intense than putting your tongue on a 9-volt battery. And we pass that current for 20 minutes, say. And then once we pass the current, this is in the cat. Once you pass anodal current in the cat, you actually get an increase in the firing frequency of those neurons underneath the electrode. How well does this translate to human? We have some data that it does translate pretty well, but it's rare to get because I rely on epilepsy patients. So let's see. So basically, what we're looking at here is the pain intensity. The greater the change in pain intensity, the greater change in the pain area of pain of the outside of the capsaicin exposure site, the better. So we saw enhanced reduction during the anodal compared to the sham. And then we also saw enhanced reduction to the anodal compared to the cathodal. There was more variability in the sham group for some reason, so that wasn't significant. But it looks like it would be significant if we had more people in the study. 25 people successfully completed this study, so it's not unreasonable to get more people. On top of that, somebody else did the same experiment, basically, using capsaicin and looking at mechanical allodynia, which is basically when you put capsaicin or you burn yourself, the area around it becomes sensitive to brush, so it feels sort of like nails on a chalkboard, or it means other pain. It's painful, but it has a weird characteristic to it as well. They found that the area was specifically reduced, and here lower arrows or lower bars are better compared to sham TDCS. So the sham TDCS, basically, we have these electrodes and we don't pass current. We basically put them on the subject's head, pass current for a very short amount of time. They think that they've had it, because I can't tell and they can't tell, and we tested that as well. But they haven't had the TDCS. So what are the potential neural mechanisms? What we found after they were stimulated in the magnet is that we had enhanced activity in medial prefrontal cortex, which is just in front of, this is also another opiate-rich area, but this is just in front of the cingulate cortex, the genu. And then we found enhanced activity in the brainstem. And interestingly enough, we found reduced activity adjacent to the electrode in the somatosensory cortex. We don't know why that is. We've never seen anything that specific before or since in the literature, really. but it is intriguing. So if we look now at a targeted analysis, looking at pregenual ACC, and then the area of periaqueductal gray, both of which release opiates, we see that the activity in response to the anodal versus the sham is greater in both the pregenual ACC and the PAG. So both of those areas are now responding better to painful stimuli. We're responding more. We don't know if it's better, but there are areas that release opiates. So, and then we see that we have suppressed activity in the left somatosensory cortex. Now, what that could be is that could be one of two things. That could either be cortical influence from the electrode, which it seems to be because that's where the electrode was, or it could be reduced activity in the spinal dorsal horn due to suppression by additional opiates. That's a longer shot. So let me see here. Where's my, there we go. Did I just go backwards? No. Why am I always have a lot of trouble with this. Let's actually just use the arrows. Okay. So this is, I promise, my last really complicated slide. What this is looking at is that areas during chronic pain that are correlated with activity in the periaqueductal gray. So the interesting thing to me is that we had left dorsolateral prefrontal cortex, the right IPL, and the parabrachial region down here. And all of those are areas that are very important in release of opiates. And so, you know, this was significant, but not significant enough, so it ended up in the supplemental. But anyway, now to completely change gears, which is basically what I do if you haven't noticed. Cross-sectional time course, these are data that we collected during COVID-19 when we were forced to all go inside, and I learned how to do survey research. So we've got one looking at greater anxiety and perceived stress after COVID-19 related worry. Those were all related to each other. We looked at this in 200 subjects. We have an additional 800 if anybody wants to analyze that data. We also had pandemic fatigue and anxiety sensitivity as measured associated factors with post-traumatic stress syndrome in Korean students, because I have a colleague that's from South Korea. And then looking also at pain prevalence and management and interference of pain among university students in South Korea. And then this latest one, which we just submitted, looking at sex influences on psychological aspects, the biopsychosocial model, which basically, to make a long story short, everybody in the medical world, the medical model of pain is that you physically injure something, it's painful. It heals, and you don't have any pain anymore. That doesn't work for most chronic pain patients. So what we look at is we also look at these biological, psychosocial, or psychological and social factors, so socioeconomic status, anxiety generally, and how those are affected. And in this case, we found that there's a relationship between how males and females how anxiety impacts their, how depression impacts their pain intensity and interference during chronic pain. So we found that they were, so the biological sex, males, basically you could explain all of their depression by saying that they also had pain, this thing called pain catastrophizing, which basically means this pain is so bad, I can't do anything about it. And there's a questionnaire that measures that. In females, both factors were equally prevalent. So depression influence made pain worse and made pain interference worse in these patients, mostly with rheumatoid arthritis.